So this is the first of several lectures on the nervous system. The nervous system is very, very, very complex. The fact that we have 3 lectures on it should tell you that. And we're only going to scratch the surface with those 3 lectures. And so the 3 lectures are divided into parts, different parts or different ways of looking at the nervous system. Today's is going to be an introduction, and we're going to talk about nervous tissues, neurons, kind of the nuts and bolts, basic science part of the nervous system, before we move on to the next 2 lectures that talk more about function, how we think, what the different parts of the brain do, things like that. This is going to talk about, at the cellular level, how does this work? And at the very beginning, we'll talk about what exactly is the nervous system. So the nervous system functions in a 3-part pattern. The nervous system always goes through these 3 steps. There's going to be some sort of an input. Those are going to come from our senses. You know our senses, the sight and smell and taste, but in feeling, in pain, and all of those different things. Our body is constantly taking in information about our surroundings and about our body through our senses. That's going to be an input. Those signals are then going to go to part of our body that is going to think about what are those sensors are telling us. That process of thinking is technically called integration. Integration is the process of taking all of the sensory inputs, putting them together, and creating some sort of a coherent thought or idea, or figuring out what is going on. What does this sensory input tell me? And then after it thinks about what does that sensory input tell me, it then has to decide, what do I do? And so it's going to have an output. In most situations, that output is going to go to a muscle. And so that is going to be a motor output. So think of it this way. You're running water to take a bath or a shower, and you don't know how hot it is. You don't want to get into a freezing cold bath. You don't want to get into a scalding hot bath. And so while the water is running, you very quickly put your finger under the running water. The senses in your finger are going to determine whether it's hot or cold, and send that signal up the nerves, up the arm, into the spinal cord, and then up to the brain. So that is the sensory input. The brain is then going to think, the signal that I'm getting, does this mean that it's hot? Does this mean that it's cold? Does it mean that it's so hot that I'm going to burn myself if I leave my hand here? What does this mean? And so based on what that means, it's going to send a signal to your hand to do something. If the water is too cold, it's probably going to tell you to pull your hand back and turn up the nozzle for the hot water. If it's too hot, it's going to tell you to very quickly pull your hand back so that you don't burn yourself, and then turn down the hot or turn up the cold. So does everybody see that repeating process? We have an input, we kind of look at what the input is and figure out what does that mean, and then say, what do I do about that? And then we send the signal to the muscles in order to react. We sense, we think, and then we react. So the nervous system is a very, very broad term. We take the nervous system and we break it down into a lot of subparts. The largest level of subparts is the central nervous system versus the peripheral nervous system. The central nervous system is made up of the brain and the spinal cord, and nothing else. Even if it is attached to the brain or attached to the spinal cord, if it is not in the brain or in the spinal cord, it is not the central nervous system. And anything that is not in the central nervous system must be in the peripheral nervous system. And then the peripheral nervous system has two branches of its own, and we just saw them. We have the sensory branch and the motor branch. And so the sensory branch of the peripheral nervous system brings information into the central nervous system over on the left. The central nervous system then sends signals back out through the motor portion of the peripheral nervous system. The sensory part always sends signals towards the central nervous system. And the motor system always sends signals away from the central nervous system. The sensory system has two subparts of its own. Both of these are senses, but one has to do with determining what's going around in our environment around us, and one of them determines what's going on inside of our body. For the somatic sensations are what you think of when you think of as our senses. The smell, the taste, the touch, all of that has to do with our surroundings. Visceral sensations would be something like hunger or a stomach ache, something like that, where it's telling you there's something wrong in our body. The motor system has the same two subparts, the somatic part and the visceral part. So the somatic part has to do with things that cause us to interact with the outside world. So think of a signal going to my hand telling it to raise up. I'm raising that hand up in order to interact with the environment around me. Whether that means waving to someone or reaching up to adjust the webcam, whatever that may be. This is usually going to go to skeletal muscle, because skeletal muscle is what causes us to move everywhere we go. The visceral is going to be some things that control our organs, like our heart and our lungs and our blood vessels and things like that. This visceral motor system is usually called the autonomic nervous system. You are not going to see the term visceral motor system very often. The autonomic nervous system has its own lecture. That's our third lecture. So we're not going to talk too much about it today. But the visceral motor system, or the autonomic nervous system, also abbreviated the ANS, has two subparts of its own. The parasympathetic nervous system and the sympathetic nervous system. The sympathetic nervous system is the fight or flight mechanism, which I'm sure you've heard of, even though if you didn't realize what exactly it was. The parasympathetic nervous system is the opposite of that. We call that rest and digest. And I'm just going to leave it at that for now. We'll talk a lot more about the autonomic nervous system in 2 weeks. So does everyone understand this general breakdown of the classification of the nervous system? Okay. So next, we're going to spend most of our time talking about the cells that make up the nervous system. Because remember, the nervous system is made up, it is an organ system. So it is made up of organs, which are made up of tissues, which are made up of cells. And so at the most basic level, the nervous system functions because the cells that make it up are doing their job. We talked about these two types of cells in an earlier lecture, where we said that the nervous system has two types of cells, neurons and neuroglia. The neurons are the nerve cells. They are the ones that carry signals from one part of the body to another. And remember, in the nervous system, those signals are electrical signals. We'll see how that works We have quite a few slides on that. In addition to the neurons, we have the neuroglia. Remember, I compared this to all of the support staff that go with an athlete or an actor or a CEO, someone who has a very, very specialized job that very few other people can do, but they have all these other people that help them to do that job. So the neuroglia support the neurons and help the neurons carry those signals. And we're going to learn about very specific neuroglia. So these are the neuroglia that we're going to learn about. These are neuroglia that you are expected to know. We're going to talk about which ones are in the CNS, which ones are in, actually, I don't know if I defined that. The central nervous system, we abbreviate CNS, and we abbreviate the peripheral nervous system PNS. So that's the abbreviations you're seeing on this slide. So astrocytes are only in the CNS, and they work to keep the chemical environment around the neurons in the correct state. We're going to see that neurons fire because the ions inside and outside of that cell are moving. And when they move, they cause electrical charges to move. And so we need to make sure that there isn't electrical noise around these neurons. We have to keep the correct concentration of ions, otherwise the neurons won't work. And so that is the job of the astrocytes. And the astrocytes are only in the central nervous system. We have something called microglia. These are they're like the dendritic or Langerhans cells that we talked about last week in the skin. These are essentially white blood cells. They're immune cells. They wander around the central nervous system looking for pathogens, and when they find them, they eat them and kill them. Another type of neuroglia are the ependymal cells. We'll talk about cerebrospinal fluid in probably lecture next week, and definitely in the lab that goes with this. So that's lab 5. So that is abbreviated CSF for cerebrospinal fluid. That is the liquid that surrounds our brain and our spinal cord. And the cells that make that cerebrospinal fluid are called ependymal cells. The next two types of neuroglia are very similar. They serve the same purpose, but they're in different locations and they have different appearances. So those are oligodendrocytes and Schwann cells. Oligodendrocytes put what we call myelin around axons in the central nervous system. Now you don't know what myelin is yet, and you don't know what axons are yet. And so we are going to leave that be until we learn about myelin and axons. But right now, notice that oligodendrocytes are in the central nervous system, and they wrap around multiple axons. Schwann cells put myelin on axons that are in the peripheral nervous system, and they wrap around one axon. Schwann cells are in the PNS, oligodendrocytes are in the CNS. Oligodendrocytes wrap around multiple axons, Schwann cells wrap around one axon. The last type of neuroglia are satellite cells. These do the same job as the astrocytes. They keep the chemical environment around the neurons correct, but they are in the peripheral nervous system, or the PNS. So here, this is a diagram showing some of the neuroglia in the central nervous system. Now there's no way to look at this diagram and say, that's the central nervous system. I'm telling you, this is the central nervous system. And so because if we know it's the central nervous system, then we can say, well, what type of neuroglia live in the CNS? And so what are these neuroglia that I'm looking at? So some of them are labeled here. So we see that the astrocytes, which keep the chemical environment correct, are right next to the neuron. The neuron is the yellow one here. It is basically anchored to it so that it stays right next to it, and it can monitor the environment around it and change it if needed. We have the microglia up here. Those were the immune cells. And ependymal cells are down here. They make the cerebrospinal fluid. And what the most important cells to see in this diagram are the oligodendrocytes. So we haven't learned about the parts of a neuron yet, but if we look at this neuron, notice that there are small branches at the top here, and then there's one very long, big branch coming out the bottom. That long branch coming out the bottom was the axon. And notice we said that the oligodendrocyte puts myelin on the axons. So that is what it's doing. It's sitting there, it's putting out multiple tentacles, kind of like an octopus, wrapping around multiple axons. Here's one that goes to that neuron. Here's one that's attached to a neuron up here somewhere. So it's attached to multiple neurons, and then it wraps around. And when it wraps around, it puts down a protective coating called myelin. Think of it like insulation on a wire. It's going to protect the electricity in the wire from getting out, and it's going to protect insulation. It's going to protect the wire inside from electrical noise on the outside. It's going to be very, very important, but we'll talk specifically about it, that there are gaps here between the myelin. That's a very important feature. But notice here the oligodendrocyte looks like an octopus with multiple tentacles reaching out, and the tentacles wrap around the axons. That is opposed to the Schwann cells in the peripheral nervous system. In the peripheral nervous system here, each of these pink rolls wrapped around the axon is a single Schwann cell. So the Schwann cell wraps its own body around the axon. The oligodendrocyte wraps a tentacle around, whereas the Schwann cell wraps its whole self around it. So here, this is the peripheral nervous system. We see the Schwann cells wrapped around the axon, and we also see some satellite cells, which again maintain the atmosphere or the environment around it, that are anchored to the neuron itself. So this is a little bit more information on that myelin. So I just said the myelin was like insulation on a wire. So here we've taken an axon and we've cut it crossways. And so you're looking kind of at a 45-degree angle, more or less straight on to an axon. That's the yellow tube here. And then we wrap myelin around. And it wraps and wraps and wraps and wraps, and it gets thicker and thicker and thicker. Now, it's not important for you to know, but that myelin is made primarily of lipids. It's kind of a fat layer insulating around it. There's no new information on this slide, and I wouldn't worry too much about the diagram or the slide, but it might help you just picture what's going on a little bit better. So, okay, so I'm going to admit, when I went through this lecture, I apparently didn't read this question, because we didn't talk about that. What type of glial cell is part of the blood-brain barrier? Well, we're gonna forget that question, okay? So I'm going to pose my own question for you, and I am going to say, if you're looking at a diagram and you see a cell that is wrapped around multiple axons, what is the name of that cell? A cell wrapped around multiple axons.
That would be an oligodendrocyte?
Yes, it's an oligodendrocyte. Is that in the central nervous system or the peripheral nervous system?
Central.
Yes, the central. And what is it putting onto the axon?
Myelin.
Yes. The oligodendrocyte is in the central nervous system. It wraps around the axon and lays down myelin. So this is our neuron. We're going to talk about the different parts of a neuron. So a neuron is a cell. Just remember that. It is a cell. All of the basic parts of a cell that we learned about are here. The nucleus, the Golgi, the mitochondria, all of that. It's here. But we're going to ignore all of that because we know that. We know all of that now. We're going to learn about the specialized parts of a neuron. So here, the yellow is our neuron. And I pointed out before that we have little short branches and then we have a long branch. The short branches in this diagram, which are up top, are called dendrites. They are going to receive a signal from another neuron. The axon sends a signal to another neuron. And so in reality, we take multiple neurons and we put them in a chain. And so we can see, it's kind of grayed out, but you can see the axon of another neuron coming into the picture here and attaching to the dendrites of this neuron. And so then there could be more dendrites down here of a third neuron. In the middle, between the dendrites and the axon, is the cell body. This is where all of that normal cell stuff happens. For a neuron, it has a special name. We call it the soma. Cell body and soma are interchangeable terms. So we have dendrites that bring in signals. Those signals go from the dendrites into the cell body and then go down and out the axon. This diagram is also pointing out the gaps between the myelin. Those gaps are called nodes of Ranvier. I'm guessing, I never remember to look it up until I'm teaching about it, but I'm guessing Ranvier is the person that discovered them. Those are going to be very, very important. We'll talk more about them later. At the very end of the axon, we have what is called the synapse. The synapse is actually the very, very, very small gap between the axon of one neuron and the dendrite of the other. They, believe it or not, don't touch each other. There's a tiny, tiny gap, and we're going to have to communicate and send a signal across that synapse. So, what part of our neuron carries the signal away from the cell body? Yes, B. It is the axon. Somebody asked, does a single neuron only have one axon? That is a very, very good question. The answer is, in most diagrams, especially in this class, it is only going to have one axon. However, in real life, there are plenty of neurons that have multiple axons, and there are actually neurons that have no axons. Think of it this way. It's a chain, right? The first neuron in that chain, there's no other neuron for it to receive a signal from. So that first one has no dendrites, but it has to send a signal to the second neuron. So it's going to have an axon. The second one is going to have dendrites and an axon. If you are the last neuron in the chain, you are the end of the line. You're not going to have an axon because you're the end of the line. There's no one to send that signal to. Some neurons will split a signal. And so in that case, there could be one dendrite and then two axons. It's going to split that signal and send it into two different places, just like if you took a wire and split it. Very good question. So when we talk about neurons, we have multiple types of neurons. And even though that they have the same parts, they all look like this. What determines what type they are is determined by what direction they are sending the signal and what type of a signal are they sending. They're not selling anything. Remember, the peripheral nervous system had the sensory portion and the motor portion. The sensory portion takes signals from the periphery, the outside of our body, and sends it towards our central nervous system. All of the neurons in that system carrying information into the central nervous system are called sensory neurons. Neurons that carry signals away from the central nervous system are called motor neurons. In most cases, it's going to be carrying those signals to muscle, in which case motor makes sense. But notice that the little definition here says it carries signals to effectors. Effectors are things that are going to carry out the command of the central nervous system. That can be muscles, but it can also be a gland. So like if you smell something really tasty and all of a sudden you start salivating. You sensed the smell. The sensory neurons carried that signal to your brain. Your brain then sends the signal to your salivary glands to create saliva. Or if you got scared, you saw something that scared you, it could send the signal to your adrenal glands to create adrenaline so that you can run away very quickly. Sensory neurons and motor neurons have another set of terms that we can use. And again, this is interchangeable. A sensory neuron is also called an afferent neuron. A motor neuron is called an efferent neuron. And I have a couple of ways that I remember which one is which. So when we send signals through a motor neuron, we're sending a signal to have an effect, right? That effect is I'm going to touch the keyboard, or I am going to change the screen brightness. And so in my mind, having an effect must go through an efferent neuron. They both start with EFF. Another way I remember this is alphabetical order. We have to have a signal go through the sensory neuron before we can have anything go through the motor neuron. An afferent, comes before E in efferent. So afferent neurons are first, efferent neurons are last. If either of those helps you remember which one is which, great. If you have a different way of remembering it, great. All I really care about is that you remember it one way or another. So, pop quiz on that already. Which one is which? Motor neurons go into or exit from the spinal cord, and are they called afferent or efferent neurons? So we got one answer. Anybody else?
D.
Yeah. So it is D. You both got it right. So motor neurons carry information away from the central nervous system. So that is exiting from the spinal cord, because the spinal cord is part of the central nervous system. And motor neurons cause an effect, and so they are efferent neurons. What time is it? We got a little bit. We'll go a little bit longer before we take a break. So something that people like, well, they don't like to get it mixed up. Something that people get mixed up fairly frequently is the difference between a neuron and a nerve. A neuron is a cell. It is that cell that we're talking about, the cell that carries an electrical signal. A nerve is not a cell. A nerve is multiple neurons with multiple axons. That the axon, another term for an axon is a nerve fiber, and blood vessels that are wrapped together in a bundle, and then they move through the body. So when somebody talks about they pinched a nerve or something like that, they're pinching this bundle of axons and blood vessels. We don't have, at least in our periphery, in our central nervous system we do, but out here in our arms and our legs, in our chest, we don't have neurons, single neurons branching all over the place. They travel in highways. And they're going down, the nerve is going down the highway, and then when one needs to exit, it exits and goes off, and the rest of them keep on going. So going down my arm is a nerve, but then the nerves near my elbow don't go the whole way. But the nerves in my fingertips are going to go the whole way. So just don't get the two mixed up. Neuron is a cell, a nerve is a larger thing made of multiple neurons and blood vessels. Actually, what slide are we on? 17. We're a little early for a break, but we're about to really change what we're talking about. So I think this is a natural time for a break. So we'll take our break now. It looks like at 6:42. We'll come back at 6:50. So about 8 minutes from now. while I was checking my email on my phone, somebody was actually thinking about science. Good for them. So the class, do sensory and motor neurons run in the same nerve bundle? And so the answer is no. Normally you're going to have a nerve, an afferent nerve, and then an efferent nerve coming back. And so it's kind of like a divided highway type system. Okay. So we talked about what the nervous system is. We talked about what a neuron looks like, what its parts are, what the different nerves, other neuroglia cells in the nervous system. The rest of our time, for the most part, is going to be spent on learning how neurons work. Okay, and so we talked about how neurons carry electrical signals. And that's a very vague idea. You probably have no idea what that means. And so we're going to try to hopefully explain that so that you can understand what we mean when we say that neurons carry electrical signals. So the first thing we need to talk about is what is going on with a neuron when it is not sending signals. When a neuron is not sending signals, we say that it has a resting membrane potential. Now, potential is an electrical term. It means a difference of charge from one side to another. And so if you measure the voltage on a battery, you measure it between the positive terminal and the negative terminal. There is a potential between them because there is a different charge on each one. One of them is positive, one of them is negative. And so when a neuron is not sending a signal, it has this resting membrane potential. And there's a difference in charge on the inside of the cell versus the outside of the cell because of the sodium-potassium pump. We see that pumped in a little more detail in a couple slides, but the sodium-potassium pump pumps potassium into the neuron and pumps potassium out of the neuron. And what's key here is it pumps 2 potassiums in and 3 sodiums out at the same time. Potassium and sodium both have a positive 1 charge. And so what's going to happen if you start with equal charges on the inside and outside of that cell, but every time that pump runs, you pump 2 positives in and 3 positives out. You're going to create an imbalance. You're going to have more positives on the outside than you do on the inside. And that is where this resting membrane potential comes from. Normally, we have positives and negatives outside, and positives and negatives on the inside. If you get rid of some of the positives on the inside, but the negatives stay behind, now we have more negatives than positives. And if that happens, the inside of the cell becomes negatively charged. When a neuron is not sending a signal, this charge is negative 70 millivolts. That is an important number to remember. The resting membrane potential of a neuron is negative 70 millivolts. Now that is not a lot of electricity, but we have multiple neurons. And so once you add them all together, we are actually talking about a fair amount of electricity. So there's several important pieces of information here. At rest, a neuron has a high concentration of sodium on the outside. Sodium is on the outside. Potassium is on the inside. Sodium out, potassium in. And because of that, because there's more sodiums on the outside than potassiums on the inside, we get a negative charge on the inside. And that negative charge is negative 70 millivolts. So in this situation, we have a bunch of sodiums on the outside and a bunch of potassiums on the inside. They want to diffuse. Remember when we talked about movement across cell membranes, we learned about diffusion. Diffusion is just the movement of things from high concentration to low concentration. So in this situation, sodium wants to come in and potassium wants to go out. That is going to be an important idea to make the neuron fire, to make it send its signal. Right now, we're at rest. The sodium can't come in, potassium can't go out. We have these channels that are closed. They're stuck where they are currently. So this resting membrane potential is just like the voltage you would measure from the positive and negative terminals on a battery. If you have a very, very, very small voltmeter and you put one probe on the outside of the cell and the other probe inside the cell, your voltmeter will actually read negative 70 millivolts. You can measure this if you have the correct instrument. And remember, this is caused by that sodium-potassium pump. Every time the pump moves, it takes 3 sodiums from the inside and pumps them out. It takes 2 potassiums from the outside and pumps them in. That's why we get high concentration of sodium on the outside and high concentration of potassium on the inside. This is a pump, remember. So this is an active transport process. It's going to require energy in the form of ATP. I don't remember the percentage, but there's some number like 30% or something. 30% of all of the energy we burn in a day goes into simply making the sodium-potassium pumps run. That's why, even if you sit in your lazy boy all day watching Netflix, you're not doing anything, you're not thinking, you're not doing anything, you're not moving, you still have to continue to eat and you still get tired. Because even though you're not doing anything, these sodium-potassium pumps constantly are running to keep the high concentration of sodium out and the high concentration of potassium in the neuron. So now we're going to make the neuron fire. And when we say fire, what we mean is we're going to make it send its electrical signal down the axon to the next neuron. And the first step here is going to be the opening of what we call a ligand-gated channel or a ligand-gated ion channel. Now, a ligand is a molecule that it's like a signal molecule that it floats around, and when it sticks to this channel, it's going to cause the channel to open. So normally the channel is closed and nothing can get through it. When the ligand binds to it, it causes it to open. And in this situation, that ligand is called acetylcholine. So the word itself is down here in the text at the bottom. We usually abbreviate acetylcholine ACH. So in the diagram, we see ACH. It is the red triangle here. It is going to float in. It's going to bind or stick to the top of that channel. That's the outside of that channel. It's going to open. And when that opens, all of a sudden, the sodium and potassium can cross that membrane and go where it wants to go. Remember, we have a whole bunch of sodium on the outside of this cell that wants to get in. And we have a whole bunch of potassium on the inside that wants to go out. But neither one of them can because the channel is closed. As soon as we open that channel, the sodium is going to come rushing in as fast as it possibly can. It wants to get equal concentrations of sodium on the inside and outside, and it wants to get our voltage from a negative to neutral. And by rushing into the cell, it can accomplish both of those. And so this process starts by the opening of a ligand-gated channel. When that ligand-gated channel opens, we said the sodium starts coming in. We were at negative 70 millivolts. Negative voltage. If we bring in sodium, sodium has a positive charge. And so our voltage is going to start increasing higher and higher and higher as that sodium comes in. We have another type of ion channel called a voltage-gated channel. These open and close not based on the binding of something like acetylcholine, but whether they are open or closed is determined by the voltage. At negative 70 millivolts, resting membrane potential, these are closed. Once the ligand-gated channel is open, we bring in enough sodium that we go from negative 70 to negative 65 to negative 60 to negative 55. And when we hit negative 50, voltage-gated channels spontaneously pop open. And we have a whole bunch of voltage-gated channels. There's a lot more voltage-gated channels than ligand-gated channels. And so when you open the voltage-gated channels, the floodgates are open. You get massive amounts of sodium coming into that cell. Lots and lots and lots of it. So much so that we actually overshoot a neutral charge. That sodium is trying to get up to 0. It's down below 0 at negative 50. It's trying to get up to 0. But there's so much sodium coming in that we actually overshoot and we go well above 0. Okay. So the change in voltage, we went from negative 70 to negative 50, and then we went from negative 50 all the way up into the positive range. That is called an action. That is called a change in potential, and we have two types. We have a graded potential, and we have an action potential. A graded potential is that small change, going from the negative 70 to the negative 50. It's small. It can be gradual. So maybe sometimes we'll go from negative 70 up to negative 60, which isn't enough to cause those voltage-gated channels to open. Enough time passes by, and then we drift back down to negative 70. We go negative 70, negative 65, negative 70, negative 60, negative 70, negative 65. We just kind of flutter around in that area. Those are graded potentials. Once we hit negative 50 and the floodgates open, we get what's called an action potential. This is an all or nothing event. We can't go up just a little bit. Every action potential is exactly the same as every other action potential. And we'll see that when we look at these diagrams here. So this is the graded potential. What you're looking at on this graph, on the x-axis we have time. So just as time goes on, we have say we have an instrument and it's monitoring our voltage over time. So we're at negative 70, we're at rest, something happens, we come up just a little bit, and then we go down, and we go up, and we go down, and we go up, and we go down. And notice some of these are bigger than others. That's a graded potential, meaning they are not all equal. It is not an all or nothing thing. There can be little ones, there can be big ones. Sometimes we hit high enough that we get the negative 50. When we hit negative 50, we're going to cause that action potential. This is showing that even though in most situations our gradient potential is going to go up towards zero, it is possible for a gradient potential to go down. That is not important to you. We're not going to talk about that in this class. You just need to know that a gradient potential is a small, temporary change that can be small or it can be big. That is in contrast to our action potential. Okay, so I think I mentioned this is the first time I've used these slides for a lecture. These slides were provided from the department. I went through and modified a fair amount, but I'm seeing a mistake that they made here. And so, on this slide with the voltage gated channels, it says that the voltage gated channels open at negative 50. And it says that's called the threshold potential. On this slide, notice they said the threshold is negative 55. On this slide, in the diagram, it says negative 55, and in the text, it says negative 50. It is, in fact, negative 55. The diagrams are correct. The text is incorrect. I apologize for that. I should have caught that. After we finish here, I'll go in and fix it for the future. So, threshold potential. The voltage that causes the voltage-gated channels to pop open is negative 55 millivolts. So we're here at resting membrane potential, negative 70. We're going along. Something happens. The ligand-gated channels open, and we come up a little bit, and we hit threshold. We hit negative 55. When we hit threshold, the voltage-gated channels pop open, and our voltage spikes big time. We see that in our graph here. We call this depolarization, and we shoot up all the way to positive 30. We start at negative 70, when we slowly increase to negative 55, and then quickly increase to positive 30. It gets up to positive 30, turns around, and now starts dropping back down. When it is dropping back down, we call that repolarization. And notice, as it drops back down, it again overshoots a bit. What we're trying to do is we're trying to reload this neuron. We're pumping the sodium back out. Sodium flooded in during depolarization. That's why our voltage went up. The positive sodium came in. We're now going to pump the sodium back out. When we do that, our voltage comes back down towards our resting membrane potential, but we actually overshoot and we go down below negative 70. When we are below negative 70, we call that hyperpolarization. So this is a very important diagram to understand. There's lots of important information here. Resting membrane potential is negative 70 millivolts. We have this slow increase to threshold potential, which is negative 55. We then get depolarization, where we shoot up. We stop at positive 30, which is another important number, positive 30 millivolts. We then repolarize. We come down below negative 70, become hyperpolarized, and then gradually come back up to level off at negative 70. Does everyone understand the steps involved here.
I have a quick question.
Go ahead.
So, when we start going up towards the negative 55,
Yes.
it goes from like 70, and then it does like a little jump, and then it does like this little plateau or something.
Mm-hmm.
Is that like the time that it takes for the voltage gates to open?
So there are a few things there. That is definitely one of them. Yes. And so there's no scale on the time here. We're talking milliseconds, if not microseconds or nanoseconds. So it is very, very fast that this is happening, but she's correct. There is a delay. Once that voltage hits, there is a tiny, tiny delay before those voltage gated channels can open. Yes.
Okay, thank you.
Other questions about this? Okay. So this is a summary of what we just talked about. So I'm not going to go through that again. We'll talk about this again at the very end of the lecture. It's not at this slide isn't at the end, but this is a very, very, very important idea, and so I'm going to continue to come back to it. That's the same thing we're just looking at. So this is how this signal moves through a neuron. So what we were just looking at was a single part, was a neuron as a whole, or a single point in the neuron. It depends. It's the same I just confused you. Let me think how I should phrase that. Let's say we're looking at a single point on the neuron there. But a neuron is big. And so when the action potential, that big peak, is happening in one place, another part of that neuron has not reached threshold yet. And so in this diagram, we're looking at it moving along a neuron. And so that action potential is going to start at the end of the axon, right where it touches the cell body. And then that action potential is going to travel down that axon away from the cell body. So if we kind of zoom out and we look at what part of that axon is currently undergoing that action potential, which one is depolarizing, which one is what part is repolarizing, and what part is doing nothing, which one is resting. So we see that here. So at the beginning, the left end here, right next to the cell body, is depolarizing. It is going up our curve. As it starts to come back down, it is now repolarizing. The part next to it is now depolarized. The furthest part of our axon is still resting. The signal hasn't gotten to it yet. And we continue to move on down the axon. So in our third step here, the first part that fired is done. It is now resting to fire again. And the depolarized portion continues to move on down the axon. Can everybody visualize this? So this is going to move down the axon. It's going to move like a bolt of lightning down the axon. It's going to start on the left, move to the right, but by the time it gets to the right end of the axon, the left end has already reset and is ready to fire again. So here's a question for you. During depolarization, is the cell becoming more positive or negative? And what ions are coming into the cell?
B.
So somebody said B. So during depolarization is going up the curve.
Yes. So it's D.
D. Sorry, I don't know if I heard B, but you might have said D. The correct answer is D as in dog. The cell is becoming more positive as it goes up the curve, and it is sodium that is coming into the cell. If we look at that curve where we come up and then down and then we hyperpolarize before we come back up the resting membrane potential, there are what we call refractory periods. Neurons fire over and over and over and over and over and over and over again very, 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 very quickly. But there are times that it cannot refire. You can't refire while you're reloading. So that is the refractory period. And we have two types of refractory period. We have the absolute refractory period and the relative refractory period. I'm going to come back to this diagram. So in order to fire again, once a neuron goes above threshold, it absolutely cannot fire again until it gets back down to threshold. So that is the absolute refractory period. When we are still above threshold, that is the absolute refractory period. There's absolutely no way that neuron can fire again. We go from the absolute refractory period to the relative refractory period. It's called the relative refractory period because it can fire again. It's just harder to make it fire again. And this is explained by this graph. Normally, we are at resting membrane potential -70. And in order to get that neuron to fire, we have to get it up to -55. We have to go up 15 millivolts. But look here in the hyperpolarized area. This is maybe we're down at -80 millivolts. And so in that case, we have to go up 25 millivolts to reach threshold. And so it's kind of if you can jump that high, go for it. But it's going to be really hard to jump that high. So it can fire in the relative refractory period, but it's going to have to take a bunch of ligand-gated channels to open in order to get a large enough voltage increase to get back up to threshold voltage. Does everyone understand the difference between absolute refractory period and relative refractory period?
So, the absolute refractory period is from the point that the line is at -55, and then to the point where it hits -55 again, like on the other side of the upside down V, I guess.
Yeah. So it would be from the peak down to where we hit -55, and then the relative refractory period is between the point where we cross -55 coming down until the point that we come back up to resting membrane potential. At first glance, it would seem that when we say we're at -56 as we're coming down, that it would be very easy to go up 1 millivolt to hit threshold again. But we have so much momentum coming down that hill that it's like trying to stop a train. That you're not going to be able to do it. Because as you're trying to increase the millivolts up by 1 at that point, the normal process has decreased it by 10. And so as we cross that, we go from the absolute refractory period to the relative refractory period.
Okay, so the only absolute refractory is only from the tip, the tip of the triangle to where it crosses the -55. This is hard for me to explain without showing you.
No. At least I think I'm picturing. Yeah. So you can see my pointer, correct?
Yeah, from there.
Yeah. From here
down to where.
To here.
Yes.
Yes.
And then everything else, the relative.
Yeah. So the relative goes from here to here,
Okay, so
where it flattens back off.
gotcha.
And then at that point, we're completely reset. We're at resting again.
Okay, that makes sense. Thank you.
You're welcome. Other questions? I know that this is a complicated concept. And so if you have questions, ask them. So there are a couple of things that determine how quickly that action potential is going to move down our axon. One of them is very simple. A wider axon is going to allow electricity to move faster than a small axon. Some axons are big, some are smaller. Think about traffic trying to move down a wide freeway versus traffic trying to move down a one-lane road. It's going to move down the freeway much faster. Same thing applies to the axon. If we have a big axon, the action potential can move quicker than if it's a small axon. The other thing that determines how quickly this moves down an axon is whether we have that myelin. Now, when we looked at the diagram at the beginning of the lecture, I implied that all axons have that myelin, but they don't. Some do, some don't. And whether it has that myelin is going to determine how quickly the electrical signal can move down the axon. If we have myelin, it moves much faster than if we don't. We use a term for the movement of the signal down a myelinated axon. That is called saltatory conduction. That's an important term. I can basically guarantee you that will come up in at least one exam. Saltatory conduction is the movement of an action potential down a myelinated axon. Now, I am not going to even try to explain how this works without a video. And I just realized I forgot to pull up the YouTube video. And so I'm going to ask your patience while I pull up this video. It's an old computer and it's doing a lot. So you're all probably looking at the videos that are on the screen here and trying to see what I watched. I found out last night my wife had never seen American Gladiators from the 90s, and so we watched American Gladiators. That's why you're seeing that, and you already know that there's fishing videos, and you did not know that I'm a Blues fan, and so there's a video of Stanley Cup from last year. So this is the video we're looking for. If you just search saltatory conduction dominoes, you'll find it. Okay, this is a very, very simple video, but it does a very good job of showing what we're trying to show here. So, can you all see the video on my screen?
I can.
Okay. So what we're looking at is we have 2 rows of dominoes. And they didn't trust themselves to not bump the table, so they have a few dominoes sitting aside so the whole thing doesn't go off on them. But what's important here is one row is domino, 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 down the whole row. In the other one, we have some dominoes, and then a domino falls and it hits a pencil, and that pencil moves and pushes another domino further down the table. And I'm going to skip ahead in the video to the point that they actually knock the dominoes down, and you're going to see what happens.
Allow the signal to travel faster. So we're going to see if we can demonstrate this.
So were you able to see the video move?
Yeah.
Okay. So what happened was when a domino fell and it hit the pencil is long. And so rather than having to knock over 6 or 7 dominoes to move 6 inches, one domino fell, and then the next domino that fell was 6 inches down. And so the side with the pencils fell quicker than the one without the dominoes. I could just find the video on your own, look it up, and watch it. So that is what's happening in our myelinated axon. What the slide says here is it allows us to skip part of the axon. The action potential moves down the axon until it hits one of those wraps of myelin. And it actually skips the myelinated section and moves on to the next node of Ranvier. And what it does is the action potential actually goes boom, 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 from one node to another, to a node, to another, to another, to another, and it skips 75% of the axon. So it allows that signal to move very, very quickly. If we don't have that myelin, it's like having an entire row of dominoes. We have to go fire here, and then here, and then here, and here, and here, and here, and here, all the way down the line until we finish that off. So saltatory conduction happens in myelinated axons and allows us to send signals faster. So now we have initiated the signal. It has moved all the way down the axon and it has gotten to the end. We now need to send that signal from the axon of one neuron to the dendrite of another neuron. And on one of our diagrams, we saw the synapse. And I said the synapse is the tiny, tiny space between an axon of one neuron and the dendrite of another. And that's what we're looking at here. In the diagram, we have the axon on top and the dendrite of the next neuron on the bottom, but they do not touch. The gap in between the axon and the dendrite is called the synaptic cleft. The synapse is this whole thing you're seeing here. The axon, the synaptic cleft, and the dendrite is the synapse. So what's going to happen is we have what we call neurotransmitters. These are little tiny molecules that are held inside vesicles. Remember, vesicles are like little bubbles inside of a cell. They're sitting there at the end of the axon waiting for the signal to be released. When that action potential moves all the way down the axon or reaches the end, it causes the neurotransmitter to get released. So it gets released into the synaptic cleft. It then floats across the synaptic cleft, binds to ligand-gated channels on the dendrite, causing the voltage to come up a little bit until it hits a threshold, and then it spikes and the next neuron fires. And so then we get that pattern repeated over and over and over again. We get an action potential that moves down the axon, causes the release of neurotransmitter, which causes ligand-gated channels to open on the next one, causes the voltage to come up, and then we spike. No, I saw this question a minute ago and forgot to answer it. So there was somebody asked a question. If you have a slight signal, like a very small peak that doesn't reach action potential, so it doesn't come up to negative 55, does the signal not get sent? And that is exactly true. What we're trying to do here is we are trying to weed out the noise. There's always electrical noise that we are not going to get a nice flat line at negative 70. We're going to kind of be fluctuating a little bit. Every time we get just a tiny little increase, we don't want the neuron to fire. We only want it to fire when we want it to fire. Otherwise, our brain is not going to work properly. Our muscles are going to be twitching and flying all over the place. We want to make sure we have a big signal before we actually spike. That's a very good question. I want to go back to the graded potential slide. So here we see some small signals. We go from negative 70 to negative 65, up to negative 60, negative 60 again, but we don't reach threshold. It's not until we hit threshold that we actually spike. And so if you zoomed in on this graph here, this would actually be kind of a wavy line with small little peaks that don't reach threshold. Very good question. So that was our synapse. And when we look at a synapse, the neuron that is at the beginning of the synapse, we call the presynaptic neuron. The neuron that receives the signal from the presynaptic neuron is called the postsynaptic neuron. Post is after, right? And pre is before. When that neurotransmitter binds to the ligand-gated channels on the postsynaptic neuron, there are actually a couple of things that can happen. It's going to open ligand-gated channels. And in most situations, that ligand-gated channel is going to allow sodium in. That's what we've been talking about up until this point. That would be considered an excitatory postsynaptic potential. We cause the voltage to come up, hit threshold, and then go. That's an excitatory potential. But there are actually some neurotransmitters that bind to ligand-gated ion channels that let in negatively charged ions. So sodium is positive. We're at negative 70. When we add positive sodiums, we come up until we hit threshold. If we open a different ion channel that allows negative ions in, we are going to go the wrong direction. We're going to cause that neuron to become hyperpolarized, just like when it was in that relative refractory state. That would be an inhibitory postsynaptic potential. If we want to prevent a neuron from firing, we can release a different neurotransmitter that's going to cause our voltage to go down. Maybe we go down to negative 80 or negative 90 or negative 100. So that way, we are so far in the basement that we can't jump up high enough to reach threshold. Okay, so that is the end of this lecture. This is most likely, conceptually, the most difficult lecture of the semester. If you understand that, you're doing really good. What I want to do is I want to back up and I want to go through that entire process again, okay? Just to make sure you understand this process. So let's say I have a neuron. It is sitting there at rest. What is its voltage? Negative 70 millivolts. And what is the term for that voltage? You have a special name. The resting membrane potential. Yeah. So we're at negative 70. Now, we are going to come up a little bit. What is going to cause our voltage to come up a little bit?
The ligand.
Yeah.
The ligand.
A ligand,
Are those-
like
are
a.
those the same as?
Go ahead.
I'm sorry. Are the ligand gated channels? Are those the same as the potassium leakage channels, or are those just those are just always open, right?
So potassium leakage channels are always open. Those were in one of the diagrams I didn't talk about, and you will not be tested on
Okay,
those.
cool.
They just
Okay.
sit there and allow some potassium to leak. That's
Okay,
all
cool.
they do. Right. So she said the ligand comes in. That ligand is our neurotransmitter, like acetylcholine or ACH. It comes in and it binds our ligand-gated ion channel. The ligand-gated ion channel now opens. When that opens, what happens?
So
Yeah. So I think you started to say it cut you off. But I think you were going to say, at least I'm going to give you benefit of the doubt, and imagine you were going to say sodium comes in. So ligand-gated channel opens, sodium comes in. Sodium comes in. Sodium has what charge?
positive.
It's positive. And so that causes our voltage to start to come up. Now it's going to come up until we hit a certain number. What is that number?
Negative 50.
Negative 55, and we call that threshold voltage. When we hit threshold voltage, our voltage is now going to spike very fast. Why? What happened when we hit threshold?
Voltage gated ion channels.
Yes. Voltage gated, when we hit threshold, we have a huge number of voltage gated channels popped open. Now we get a whole bunch of sodium coming in, and our voltage goes up. We have a term for that period of time when our voltage is going up. What is that called? Depolarization. So we're going to depolarize until we hit the top of the peak. At what voltage do we stop going up?
Positive 30.
Yeah, very good. The top of the peak is positive 30. And now we start to come back down. What do we call it when we're coming back down?
Repolarization.
Repolarization. And what is happening to bring our voltage back down? So I realizing we may not have talked too much about this. And so the voltage went up because sodium came in. And so if we want the voltage to go back down, what do you think we have to do?
Push it out.
Push it out. The sodium potassium pump has to take a little bit of time to pump that sodium back out. So that's what's happening when we're repolarizing. The sodium is getting pumped out. So we peaked at positive 30. What do we come down to? What voltage do we come down to?
Just below negative 70, just a little bit farther than resting.
Yeah. So we come down below resting, below negative 70. There's no defined specific number for that. We said at that point we were hyperpolarized. We were down below resting, and then we gradually, slowly come back up to resting. So that is the steps of an action potential. So if you can walk yourself through that, you have understood the most complicated parts here. There's two other important ideas here other than the parts of the neuron. That's just memorization. So I'm not going to recap that. So this action potential moves down an axon. What happens when it reaches the end of an axon? What happens?
Like at the end of the axon, it'll hit the neurotransmitter response.
Yeah.
Signal those first.
Yep. When the action potential reaches the end of the axon, it causes the release of neurotransmitter, which floats across the synapse, binding to the ligand-gated ion channel on the next neuron, and now we're right back where we started. And so it's a cycle, and it goes from neuron to neuron to neuron. The last important concept is easier. At rest, is there more sodium inside or outside of a cell?
Outside.
Outside. At rest, is there more potassium inside or outside of a cell?
Inside.
Inside, yes. Thank you. So that's it. If you can remember, the sodium is outside, potassium is inside. Walk your way through that action potential curve, knowing what the terms are called and why the graph is doing what it's doing, and then understand how that works in a neuron, moving down the axon, releasing neurotransmitter to restart the cycle on the next one. You should be golden. If you can understand that, you understand how this works. Okay, so now I am done. Any questions? Okay. As always, if you do have questions, let me know. I'm happy to answer them. And remember, you have 2 exams due by the next time we meet. And please don't wait until the last day. If you wait until the last day, you're asking for something bad to happen. Don't wait until the last day. Anything... Is it a neurotransmitter that causes the initial neuron to fire? Another good question. So what he's asking is, so I kind of ignored the first neuron in the line, right? I talked about neurons in a line, and so it fires when it receives a signal from the first one. But there has to be a first one. And so what causes that first neurotransmitter to fire depends on where it is. And so in the sensory nervous system, that first neuron is going to be someplace like the tip of my finger. So what actually happens, the way you feel touch, is when you push on something, we have what we call pressure sensitive ion channels. And so when you push, rather than a ligand gated ion channel, the pressure gated ion channel opens the harder you push. So then you get that first little bit, and then you hit threshold and you go. Another example is in your eyes. When the light hits the first neuron on your retina, that light actually gets converted into a voltage change, which causes it to hit threshold and fire. If you are in the motor system, the first neuron in the line is in your central nervous system, your brain or your spinal cord. And in that situation, either let me back up. I'm making it more complicated than it is. If you're in the central nervous system, it will be a neurotransmitter. Because you're going to have a neuron that does the integration, the thinking part, and it is going to send a neurotransmitter to the first neuron in the motor line to start that signal. Other questions? Okay. So if you have questions, let me know. I can answer by email. If you want me to get on and walk through these things with you again, I'm happy to do that. Just let me know. We'll find a time. Otherwise, I hope you have a good week. Good luck on your exam. If you're in this live class, just one of your classmates is looking for study partners, and just put some contact information in the group chat if you want to look for someone to study with. Okay, so I will see you next week. Good luck with your exam.
Thank you. Have a good night.
You too.